The overall goal of this electroencephalographic, heart rate, and galvanic skin response assessment is to study and measure the unconscious neurophysiological perception of advertising, and particularly of public service announcements.
This method can help answer key questions in the neuroscience field, such as the assessment of the cognitive and emotional reaction to advertising, as well as its impact in the population subgroups.
The main advantage of this technique is that it enables the measurements of neurophysiological unconscious reactions to the stimuli and obtains significant results even with a number of participants lower than that required in traditional marketing researches.
Describing and illustrating the procedure will be Anton Maglione and Enrica Modica, which are two bioengineering researchers in my lab, as well as Myriam Caratù, which is a marketing and communication researcher, as well as Patrizia Cherubino, which is neuromarketing researcher in my lab.
Begin by escorting the participant to a chair in front of a computer screen. Use a chlorhexidine antiseptic solution to disinfect and remove the most superficial lipid layer of the skin on the forehead and on the earlobes. Next, apply a 6th dry electrode electroencephalography or EEG band to the participant's forehead according to the 1020 International System. Then, place the reference and the ground electrode, one on each earlobe. Acquire the EEG signal through an EEG amplifier system and a related